Welcome everyone. Have you ever wondered how the intricate structures in our bodies, like the folds in our brain or the villi in our intestines, come to be? What influences these patterns and shapes during development? Today, we're going to explore a fascinating scientific study that delves into these questions, focusing on a process called epithelial folding and the role of environmental asymmetry in this process. Let's dive in and discover the intriguing findings of this research. Epithelial folding is a fundamental process in morphogenesis, which is the biological process that enables an organism to develop its shape. This phenomenon is universal and can be observed in various biological structures. For instance, the folds in our brain, known as gyri, the villi in our intestinal tract, and even the special structures used by insect larvae to pupate, all form through epithelial folding. This process plays a crucial role in shaping the structures of our bodies. Today, we will delve into a study that explores how environmental factors can influence this fascinating process. In this study, the researchers focused on the role of environmental asymmetry in epithelial folding. They looked at the constraints that the environment places on the epithelial sheet, specifically the asymmetry of these constraints. The degree of asymmetry is a measure of whether the basal or apical side of the epithelium is closer to the constraint wall. This asymmetry can influence how the epithelial sheet folds and develops. Let's look at how they investigated this relationship. The researchers used a method called three-dimensional vertex simulations to investigate the relationship between the degree of asymmetry and epithelial folding morphology. This method allows for a detailed analysis of how changes in the environment can influence the folding patterns of the epithelial sheets. The results from the simulations were quite fascinating. As the degree of asymmetry changed, so did the folding patterns of the epithelial sheets. They observed a progression from spot patterns to labyrinth patterns and then to hole patterns. This indicates that the environmental asymmetry has a significant impact on the folding patterns of the epithelial sheets. In this study, an energy functional of the cell sheet was developed to discuss pattern formation. This was in response to buckling caused by compression due to cell proliferation under periodic boundary conditions. The researchers considered the cell sheet being compressed by force N from the x and y directions. They derived the Swift-Hohenberg equation from the overdamped relaxation dynamics of the displacement field W. This equation confirmed the transition from a dot pattern to a hole pattern via a labyrinth pattern with increasing degree of asymmetry. Upon examining the stability of the solution where W equals 0, Laplacian and the square of Laplacian operators were found as instability factors. However, the terms with the offset ramp functions in the equation suppress the amplitude of W. In conclusion, the degree of asymmetry was identified as the parameter for pattern selection, providing a deeper understanding of how environmental asymmetry influences epithelial folding. Our exploration today has underscored that the physical environment surrounding the epithelial sheet, especially its degree of asymmetry, significantly influences the folding of the sheet. This suggests that nature might be using environmental asymmetry as a tool to create functionally diverse folding structures. We see this in the diverse patterns such as the labyrinth-style gyri in the brain and the spot-style villi in the intestines. Understanding this could open up new possibilities in fields like tissue engineering and regenerative medicine, where controlling tissue shape and structure is a key challenge.